So last time we said that the action potential is caused by an influx of sodium ions into the cell. So that positive current of sodium ions coming into the cell is what drives the membrane potential up during the rising phase of the action potential. And so now we're going to talk about how that happens. So those sodium ions come into the cell through these voltage gated sodium channels. These are proteins in the membrane, and you can kind of see what they look like here. So each one is a single polypeptide, a single protein that crosses the membrane several times. There are basically 4 domains that make up the actual channel or the pore, the part that goes through the membrane and lets that the sodium ions actually interact with. And the pore loop here, this red part of each of those domains, is also the selectivity filter. So that's what determines which ions can go through. So it only lets sodium through and not, for example, potassium. Partly the reason, the main way that the channel can tell the difference between sodium and potassium is size. So sodium ions are smaller than potassium ions, especially when they're surrounded by water molecules. So water molecules, remember, are attracted to ions. They form basically a shell around each ion. And so with a water molecule attached, the sodium ions fit right through the selectivity filter, whereas potassium ions do not. And so, and of course, a negatively charged ion also would not be attracted to the pore loop. So that's what determines which ions go through. But the part that determines whether or not the channel actually opens consists of these regions here, this part of the protein called the voltage sensor. And the voltage sensor is kind of what it sounds like. It's a group of amino acids, and so you can see within each domain there's one loop that has that's kind of colored purple here. So there's one here, one here, one here, and one here. So you have to picture all 4 of those surrounding the pore in a ring. And those amino acids have positively charged side chains. And what that means is that at the neutral pH, they have an extra proton. So that extra proton gives them a net charge of +1, which means that they are pulled inward to the cell when the cell membrane has a negative membrane potential. So at, say, -65, for example, the negative charge inside the cell kind of pulls these ions down or pulls these amino acids down. And what that does is it causes this part of the protein, the gated part, to be shaped like this in such a way that basically the channel is closed. And then when the membrane becomes less negative, it doesn't have to go all the way to positive, but if it goes from -65 to around -40 or so, and the exact threshold can vary from one channel to the next or one type of cell to the next. But typical channels like this have a threshold of -40, but that slightly less negative membrane potential is enough to sort of release the inward pull on these positively charged amino acids, and that lets them go up. And when they do that, they kind of pull the rest of the protein with it, and that then means that now this channel is open. So sodium ions are free to come in. And so that is the voltage sensor. That is why we call it a voltage gated sodium channel, because it is closed when the membrane is below that threshold potential, -40 or so, and it opens when it gets above -40. But they're a little more complicated than that. There are actually really 3 different states that these channels can be in. Again, they start off closed. So when the membrane is at rest, again, that's about -65 millivolts, give or take, the channels are closed. That means no ions can go through. But once the membrane gets to the threshold potential, about -40, they basically instantly open. So up here, you can actually see, here's the membrane potential, and then down here, these are measuring currents through individual channels. So you can do this with ion channels there's a technique called patch clamp recording, where you take a very, very tiny electrode. Again, the electrode is basically just a long, skinny glass pipette. Put it up to the surface of a cell membrane, and you can actually pull a little chunk of the membrane off such that it is basically sealed against that piece of glass. And then you can, if there happens to be an ion channel under that tip where you pulled off, you can record the activity of that single channel. Obviously, this requires really high-tech equipment and a lot of practice, but that's what these recordings here were made from. So each of these little traces is the recording from an individual sodium channel. So you can see the current. Again, inward current is negative, so sodium ions coming in represents a negative current. So that's why you see the current going down when the channel is open. But what's important is that when the channel opens, again, you've got, we're looking at a time scale here of less than a millisecond. So the whole from here to say here, so that's just 5 milliseconds. So in just a fraction of a millisecond, these channels open. Now there's a little bit of variation, so some open slightly later than others. But again, within a millisecond, all three of these different channels all open, and they open basically right away. So they go from no current straight to some current, and the amount of current really depends, remember, on the difference in membrane potential between the membrane potential and the equilibrium potential. But the point is that they open right away, and then they stay open for about a millisecond. So each of these during its open phase stays open for roughly a millisecond, maybe longer. Again, these are mostly sort of random movements of these proteins that ultimately are sort of leading to these changes. So there's always going to be variation comparing one channel to another. Even if you look at the same channel twice, it might behave slightly differently from one moment to another. But on average, about 1 millisecond is how long the current lasts. And then after that millisecond, the channels enter a new state called inactivation. So this basically is what the channel looks like when it's closed. And then after crossing threshold, it goes into this state, which we call the open state. And then about a millisecond after opening, another voltage gated or electrically voltage sensitive part of the protein kind of swings into place. This little purple tail thing swings here into the pore. Even though the membrane is still negative, it swings in there, and that blocks the channel. And so we call this state inactivated. So after about a millisecond of being opened, the channel becomes inactivated. And that's basically the same thing as being closed, because no ions can go through. But what's important is that it will stay closed even if the membrane is above threshold. So you notice here, we've left our membrane potential at -40 all the way down here. So it's staying at -40, and so that's well above threshold for several milliseconds. But these channels do not let ions go through, because again, from this point on, they are inactivated. So we call this part inactivated. And that means that not only will they not let ions go through, but they cannot be opened again until the membrane goes back down below threshold. So once they get to back down below threshold, then they go back to basically the closed state again. So closed, open, and inactivated. In fact, sometimes this process of going from inactivated back to closed we call de-inactivation, which is kind of a mouthful and sounds like a kind of a double negative, kind of a self-contradictory term, but that is what that process is sometimes referred to. Now, so again, these are the channels that drive or allow the sodium ions to enter the cell during that rising phase. And the reason, in fact, the rising phase stops is because of that de-inactivation. So we'll come back and look at that process more in a minute. Just a couple more things about these channels. There are lots of substances that can block these channels. That's true of a lot of proteins. So there are most proteins, most receptors and ion channels and proteins in cells have chemicals that interact with them that are not normally found in the body. In fact, that's how most drugs and medications work, is by a molecule that binds to a receptor or binds to an enzyme or binds to an ion channel and affects the cell in some way that it would not normally be affected by. And so there are lots of chemicals that block these sodium channels that are either used in research or kind of important otherwise. So one important one is this stuff here called tetrodotoxin, or usually just called TTX. So TTX and all these drugs, what they do is they block the channel. That means that even when the channel is open, sodium ions can't go through because the drug just happens to fit somewhere in this pore, or it somehow prevents the channel from opening. And so TTX works that way. TTX is actually found in the poison gland of some fish, especially pufferfish. So the pufferfish poison gland has this TTX substance in it. So this is a self-defense mechanism for the fish, because if an animal eats it, then TTX enters the sodium channels in the nerves, but also in the muscle tissue. So muscles, we'll learn, also can produce action potentials, not just nerves or not just neurons. So in the muscles that control breathing, or the heart, for example, this poison will stop those muscles from being able to contract, because the action potential is necessary to cause muscle contraction. And that's that we usually kill whoever tries to eat one of these fish. In fact, there is a delicacy of these pufferfish called fugu. That's the Japanese word, and it is served in some sushi restaurants. But it's very difficult to serve, because you have to very carefully cut out the poison gland, because otherwise you can kill your customers. So in fact, in Japan, in countries where these fish are served, it can only be served in a restaurant that's heavily regulated by the government. Someone has to come in and inspect, and then the poison glands can't be thrown away. They have to be put in a special biohazard box with a lock on it. Someone has to collect it. So it's a very potent poison. It's also really important in research. It's used in research labs to study action potentials or study electrophysiology of neurons, because you can apply the toxin to a neuron in culture, for example, and essentially by shutting off these voltage-gated sodium channels, you can stop action potentials from happening, and then you can look at how membrane potentials and other channels affect or behave in the absence of action potentials. This stuff over here, saxitoxin, is produced by certain types of algae, and normally it doesn't cause much of a problem, but sometimes these algae will kind of grow and reproduce out of control, and some of them, when they form these big blooms of offspring, it has kind of a reddish brownish color to the water. And so sometimes when these algae that produce this toxin kind of grow out of control, it's called red tide poisoning. So if you've ever heard of red tide poisoning, or red tide, that's where that term comes from. So red tide poisoning then is when fish and shellfish, other animals in the water, eat these algae, or they get in their diet, and so this saxitoxin builds up, which can kill off sea life, but it can also affect people, because if you eat a fish or go swimming during a red tide, you can also get very sick. One thing that's relevant here is that these blooms are becoming more common, because they are one, affected by climate change, but more importantly, they also are affected by runoff from rivers. So in places where you have a lot of agricultural land, fertilizers from farmland can, if they're not carefully applied or carefully monitored, can end up in freshwater supplies, which eventually make their way down to the ocean, and then those can trigger these red tides. And so that's just a link between kind of human activity and these kind of poisons. One example of a sodium channel blocker that you may have encountered also is lidocaine. So if you've ever been to the dentist, to get a cavity filled or any other kind of procedure, lidocaine will block these channels, and so it's injected usually into the tissue around where, you know, maybe you're having dental work done or any other kind of sort of superficial surgery. And what it does is it prevents action potentials from traveling along the axons that are carrying pain and other sensory information to the brain. So that's why it essentially numbs the skin and the muscle tissue and teeth or whatever tissue it's injected into. And so, you know, different drugs that all interact with the same protein but have different effects because they maybe bind in different ways or have different impacts on the channel itself. And then you can also have activators of these channels. So these are molecules that prevent the channel from closing, which means that they can't either close or inactivate it. So the channels stay open after an action potential has begun and do not close. And that's just as bad as not being able to open, because if they can't close then or inactivate, then that means the membrane cannot return to resting potential, which means another action potential can't be generated. And so it has in the same on the organism, it has basically the same effect. It can these activators can block the muscles and nerves that control breathing and heart rate. This stuff, batrachotoxin, is found in the skin of some frogs, especially these guys, the poison dart frogs. These are native, a lot of them, to the rainforests of South America, and some of the indigenous tribes that live in that area actually use these frogs. They kill them, and then they sort of grind their skin or grind their poison glands onto their arrows, on the tips of their arrows, and use them in hunting. So when they're hunting in the forest, they have this poison on the tips of their arrows, and that kills their prey. In fact, that's why these frogs are called poison dart frogs, is because their skin is used for that purpose. Now, for the frog, of course, again, it's a self-defense mechanism. If another animal eats one, it will die. That's why they're actually really brightly colored. Usually, when you see these frogs, they have bright yellow, green, or red skin, and you know it's very bad camouflage, but it's a warning sign to predators: hey, don't eat me because you'll die. And then here's another example. This is from a plant. So there's actually several plant species that produce sodium channel activators. This one's veratridine, comes from lilies. This is one of the reasons why you shouldn't eat houseplants, or let your pets eat houseplants, because this is one of the things that can make them very, very sick. And again, all of these are also important for research. So if you study sodium channels or study electrophysiology of neurons, you might use these drugs to have different effects on the cell. Sometimes, you know, like I mentioned, lidocaine, some of these kinds of molecules too can lead to drugs that might help treat diseases also. So now let's go back and look at the action potential in more detail and look at the parts and see what's going on now that we know kind of what's going on at the membrane, how we can explain these different properties. So first, let's talk about the threshold. So the threshold is that minimum depolarization necessary for the action potential to start, and that threshold is determined by the sodium channel. This notation, by the way, is Na sub V, is a notation that is used to refer to sodium channels, proteins that are sodium channels. And so these channels have a threshold. The threshold can vary slightly from one type of channel to the next, and there is more than one type of voltage gated sodium channel, and they can vary in threshold, they can vary in opening time, and all these other factors. But minus 40 is a typical threshold. But as long as the membrane potential is below minus 40, it's a lot more negative than minus 40, then the channels will stay closed. So then the rising phase of the action potential refers to once you've passed threshold and start opening those voltage gated sodium channels, the membrane potential starts to go up, because again, you're letting sodium into the cell at a very high rate. The current for sodium is very large at this point because there's such a big difference between the equilibrium potential for sodium and the membrane potential. That's this distance here. So again, for a typical cell, the membrane potential is at rest about minus 60 something, and then for sodium the equilibrium potential is close to plus 60 something. So that's a difference of more than 120 millivolts between those two numbers. And so that tells you the magnitude of the current that you see during the rising phase. You have the other factor that determines current is the number of open channels. So if you have a whole bunch of channels opening at once and that big difference in potential, you're going to see a large current of sodium. And so that's what's happening during the rising phase. The rising phase essentially continues on past zero. So even as the inside of the cell becomes positive relative to the outside, you still have sodium rushing out, because again, we're not yet to the equilibrium potential of sodium. So if you don't close or inactivate those sodium channels, this overshoot phase, now we're in the overshoot, will continue until you get to the equilibrium potential for sodium, until you get to plus 65 millivolts. But before that happens, you enter the falling phase, because here what happens is those sodium channels become inactivated. So as the sodium channels inactivate, the sodium current stops and the potassium current takes over. So remember, before we even started this, we had open potassium channels. They're those potassium leak channels. So as soon as the sodium channels close, the sodium or the potassium current kind of reasserts itself. And so potassium now we're at a point where the difference, the potassium current again is a function of the conductance for potassium, which is a property of how many channels are open, but also this distance between now what the membrane potential is. Now we're up to maybe plus 40, plus 50, whatever it got to, and then the equilibrium potential for potassium, which is way down here. So now you have a difference in potential of, again, probably more than 100 millivolts, and so that will create a very large, relatively large potassium current. But again, because potassium concentration is higher on the inside of the cell, the potassium current will be outward, positive, and therefore it will bring the membrane potential down. And so that's why we call this the falling phase. And so potassium leaves. Now, one of the things that I haven't mentioned is that there are also voltage-gated potassium channels. So voltage-gated potassium channels actually do the same thing essentially as voltage-gated sodium channels. The only important difference is that they are open slightly later. They don't open quite as quickly as the sodium channels. So they're triggered by that depolarization past the threshold, and the threshold's about the same for both sodium and potassium, but the timing is different. So it takes the potassium channels slightly longer to open, so they don't take effect quite as quickly as the sodium channels. In fact, that's why the falling phase is as rapid as it is. So it's not just those potassium leak channels, it's additional voltage-gated potassium channels that are open. And in fact, those voltage-gated potassium channels also explain the undershoot phase, because now we're at the undershoot, which means, again, we're down below what was resting potential. And the reason is that now we're at a point where, again, the sodium channels are still closed, because now they're below threshold, but now they've gone from being inactivated to being just plain closed. And then once you get down past minus 40, the voltage-gated potassium channels also start to close, but there's a window in here where you have extra potassium channels open, and so that means the permeability for potassium is open. And so if you remember back to the Goldman equation, that means that the membrane potential will drift further toward the equilibrium potential for potassium, and that's what happens. So during the undershoot, we get closer to the equilibrium potential for potassium. Again, it doesn't quite get there because, one, there are still some sodium channels that might be open, and there are other ion channels that affect the membrane potential, and those voltage-gated potassium channels are starting to close because, again, they are also below their threshold. But just like the opening, they have a delayed closing, so they take a little bit longer to close. And as they close, the membrane potential relatively gradually drifts back toward the resting potential. I say relatively because, again, this whole thing took about 2 milliseconds. So from the moment that you cross threshold to the moment that you get back to rest, you're talking about maybe 1.5 milliseconds. So it's a very, very short process. Here you can see what's actually going on at the membrane with the different currents. So again, during the rising phase, that's dominated by sodium influx. So you have sodium coming into the cell, and here you can actually see the currents through individual voltage gated sodium channels. Like I said, some of them open slightly later, some of them may stay open slightly longer, but about a millisecond or so of time, you have all these sodium channels open, and so the total sodium current will be the sum of all these individual channels open simultaneously. And then down here, you can see current through potassium channels. So again, the potassium channels open, but their opening is delayed. So they don't really start opening until most of these sodium channels have already closed or have started to close. And that means that the total sodium current, really the sum of those, potassium currents, doesn't really become a factor until after the sodium channels are closed. And so after the sodium current has stopped, the potassium efflux, the potassium leaving the cell, is what really dominates. So the two together kind of shows you the current curve. So again, during the rising phase, you have inward sodium current, and then during the falling phase, you have outward potassium current. And so that's just what's happening during those phases. Now, there's two other things to mention about the phases of the action potential, and I briefly mentioned them earlier on, and that is that there's a limit to how many action potentials you can have in a given amount of time. That's because you have to wait for one action potential to finish before the second one can begin. So that there's a period of time from the initiation of the action potential, basically from the moment it crosses threshold, to the point where it gets back to threshold, where no matter what you do, you cannot create another action potential. We call that the absolute refractory period. So the absolute refractory period is defined by when those sodium channels are able to be opened again. Again, they cannot be opened until the membrane potential gets down below that threshold, or what we say is that the channels have to be de-inactivated in order to hypothetically open again. So that time frame, which again maybe a millisecond, 1.5 milliseconds from initiation, is the kind of the upper limit on how many action potentials you can have. So about one every millisecond, or one every 1.5 milliseconds, is the maximum firing rate of a hypothetical firing rate of a neuron. Again, in practice, neurons rarely fire that fast, but you can artificially force action potential generation at that rate if you want. But there is another period, even after the absolute refractory period, where there's still you can generate more action potentials, but it takes a little more work. We call this the relative refractory period. So during the relative refractory period, you can generate another action potential. So if the membrane potential is above threshold, those sodium channels are ready to start another action potential. But it's during this undershoot phase, which means that the membrane potential is again below rest. So let's assume that minus 40 is your threshold. So now, instead of having to get from here to initiate action potential, you have to get from here to here. So this extra distance here is necessary, or this extra amount of depolarization is necessary if you want to start a new action potential.